Welcome to the lecture video for Chapter 4, Part 3. This is the second part of eukaryotic cells. We're going to look at the cytoskeleton and then the composition of the cytoskeleton, the centrioles and their role in the cell, especially the mitotic spindle, flagella and cilia, which are organelles of locomotion located on the outside of eukaryotic cells. And then we're going to look at the animal cell glycocalyx and the plant cell wall and look at the junctions and structures that hold cells together to build tissues. And plasmodesmata is a special structure in the plant cell wall. The cytoskeleton is really important to a eukaryotic cell. There's a pseudocytoskeleton in prokaryotic cells that only exists for a temporary period of time to help push plasmids to new daughter cells when the cell divides. But in the eukaryotic cell, remember, eukaryotic cell is like thousands of times bigger than a prokaryotic cell. And so to keep the cell from flattening out, you need the cytoskeleton inside to help maintain the shape. And what it is these microtubules and intermediate filaments and microfilaments that are crisscrossing throughout the cell, helping to push opposite sides of the cell apart. This also holds some of the organelles in a specific position, and it allows the movement of cytoplasm and structures, vesicles, or small organelles around within the cell. And if there is cilia and flagella on the outside of the cell for movement, it allows the cytoskeleton to anchor to those through the cell membrane for more support. Cytoskeleton is composed of three main parts: the microfilaments, intermediate filaments, and microtubules. And they're all very different from each other, and they have very different functions. Microfilaments are made of actin subunits that are linked together in long chains, and they're associated with movement in the cell, either the whole cell to move or just parts within the cell. They also help determine and stabilize the shape of the cell. The intermediate filaments are the most diverse group of cytoskeleton structures, elements. These are several strands of a fibrous protein that are wound together. Fibrous proteins they don't dissolve in water. They're very, very strong. So they include things like collagen and elastin that make up your cartilage and your ligaments and your tendons. This includes the keratin that makes up your fingernail and your hair. And so these are really, really strong fibers. And their function is all structural. They bear tension. They're really good at taking on a lot of stress. They maintain the cell shape, and then they anchor the nucleus and other organelles in place. We saw in a previous lecture how the nucleus is right next to the endoplasmic reticulums, which is very close to the Golgi, and it's because of the intermediate filaments holding these organelles close together that keeps them aligned, which allows them to be very effective in manufacturing and distributing things within the cell. Microtubules help form a rigid internal skeleton. Not all cells are going to have as many. They're formed of tubulin subunits, which coil around, and you can kind of see where the tubulin's a dimer made of two different polypeptide chains. And then it's kind of, as it links together, it forms this hollow tube. Okay. This also provides a framework for motor proteins to allow them to move structures within the cell. Motor proteins are these special proteins that will lock onto the microtubule with one end, and they'll lock onto a vesicle or an organelle or a large molecule with the other, and they can physically move themselves along the microtubule to move something from one place in the cell to another. Centrosomes are a structure that's really important to eukaryotic cells because this is what we use to build the mitotic spindle. We're going to talk about mitosis or asexual cellular reproduction in a later chapter. But the purpose of the spindle is to make sure that when we copy the chromosomes, we get one copy of each chromosome in each new daughter cell. And this is a really complex process to ensure that this occurs, because if it doesn't happen, if we get two copies of a chromosome in one cell and nothing in the other, there's a good chance the cells won't survive, and we just wasted all our time and energy reproducing. It's composed of 9 triplets of microtubules, and you can see, the triplet, there's a triplet, 2, 3, 4, 5, 6, 7, 8, 9. And then in between those are non-tubulin proteins that help to hold them together. Okay, so this is a really, really strong structure. There's two of these that run at right angles to each other. And again, most of the time in the cell, the centrosome is, and the 2 centrioles are going to be inactive. It isn't going to be until we're ready for the cell to replicate that they really take on their function. Cilia and flagella are microtubule structures that are outside of the cell. And when they move, or when the microtubules bend, it causes those to move, which causes the cell to move. Flagella are longer, have a very whip-like motion because there's a power stroke and then a recovery stroke, kind of like you were rowing a boat or paddling a canoe, where you have a power stroke and then a recovery stroke. Cilia tend to be shorter, more numerous, and they beat in a synchronized fashion. And we find cilia lining our respiratory passageways, where they help to sweep mucus and debris up and out of our lungs and out of our bronchioles and other airway tubes. These are sperm, and you can see the long, thin flagella that allows the sperm to swim. They have this characteristic 9+2 array of microtubules, and if you look at it in cross-section, you see there are 9 pairs around the perimeter, and then there are 2 in the middle that are not fused to each other. And when there's a small piece of protein called dynin that's between these sets of microtubules, and when that dynin contracts, it causes the whip-like power stroke, and then the recovery stroke ensues. And so this is what allows movement in eukaryotic cells. Extracellularly, of course, plant has a plant cell wall, and we looked at that in detail in a previous lecture. And the thing about it is, when plant cells reproduce, they're likely to stay firmly attached to another plant cell. And what happens is, when the plant cell divides and it builds new cell wall between the 2 daughter cells, it builds this, what's called the middle lamella. It's a thin layer of mostly pectin that is built between the 2 plant cells. And what it does is it holds those cells together very tightly. And plant tissue is likely to remain bound together very, very tightly. Think about wood. The strength of wood is largely because of that cell wall and the middle lamella. This is a barrier to infection. It's hard for viruses to get through. Other things that can infect the plant cell, unless you run amok with a weed whip or an insect chews or bites through the cell wall to create a portal, the plant's pretty well protected against disease. And of course, it's there for support. You can get that high turgor pressure on the inside of the cell pushing against the cell wall, and that makes plant cells very, very strong. Animal cells, if they're going to stay connected to each other, it's not going to be as tight a connection because the animals need to move. And so they're going to have this glycocalyx on the outside, which we've kind of looked at before, with long collagen fibers and other fibrous proteins. And then they're going to have glycoproteins and proteoglycans, which are going to be attached to the plasma membrane and then kind of weaving in amongst the collagen fibers. And then there's going to be linking proteins, which are the fibronectin, which hold everything to the integrins, or the proteins that are in the cell membrane that bind to the glycocalyx. And then those integrins can also be bound to the cytoskeleton on the inside. And so what this does, it allows this glycocalyx to interact with the glycocalyx of another cell, and some bonding can occur to help to develop tissues. If we really want to build tissues, though, in animal cells, we're gonna build junctions. And there are 3 different junctions that occur in animal cells: a tight junction, desmosomes or anchoring junctions, and gap junctions. We're going to start with the gap junction because it very closely resembles a similar structure in plant cells. Plant cells don't have junctions because of the middle lamella, and the strong cell wall is anchoring the cells together so tightly. But plant cells need to be able to move material from cell to cell. They need to be able to allow water to move from the roots up through the stem to the leaves for photosynthesis, and they need to allow sugars that are made in photosynthesis to pass back down through the plant tissues to all the various structures in the cell that need the sugars to make energy to survive. And so these pathways from cell to cell are called plasmodesmata. Plasmodesma for singular, plasmodesmata plural. And it's basically a channel between the two cells. The smooth endoplasmic reticulum creates the borders of this channel, and then it allows cytoplasm and other materials to freely flow from one cell to the other. The structure in animal cells that's similar to that is called a gap junction. And what happens in a gap junction is you have the proteins that form a structure called a connexon, which is six smaller proteins bound together, and when they relax and open, they create a pore between two cells. So here's one cell up here, here's another cell down here, and here's the connexons or gap junctions holding the two together. And when it opens, now cytoplasm and small ions and small molecules can freely flow from one cell to the other. Tight junctions are waterproof seals between animal cells. Now, this is why when you soak in a bathtub or you go swimming, you don't gain weight you don't bloat with water. And that's because your tight junctions are waterproof. They don't allow water to pass between the cells. And they're very, very strong. And of course, this is necessary. Otherwise, as we moved, we would just tear apart easily. These are especially found in the epithelial cells that line your internal organs in your cavity. So all your blood vessels, your gastrointestinal tract, your urogenital tract, all your major organs are going to have lots and lots of tight junctions. Desmosomes are not as strong as a tight junction. They're not waterproof. Water can pass through. But they're still fairly strong in tissues that need to stretch. So like your heart, your lungs, your muscles, we don't really want a lot of tight junctions there because it would hinder the movement of those organs or tissues. And so instead we have desmosomes, which used to be called anchoring junctions. And these have a series of short proteins called cadherins that are within the plasma membrane, and they kind of act like your book calls them spot welds. And if you've ever seen somebody who's welding, if they're putting two big pieces together, what they do first is kind of just create a couple of little spots where they touch the metal between the two pieces of metal to briefly tag them together, and then they'll fill in the rest of the weld later. And desmosomes are kind of like that. There aren't a lot of them. It's not like you have a solid barrier of them. You just have enough of them to keep the cells from coming apart when they move. So if we compare the animal cell junctions, our tight junction, waterproof, going to keep water from escaping. So all of your blood vessels, your organs, any place where there's fluids, we don't want them to leave from one cell to the other or to get between the cells. The anchoring junctions are going to be in tissues that need to move. So it holds them together, but not nearly as tightly as a tight junction. And then the gap junctions create pores between cells that allow for parts of the cytoplasm to flow from one cell to the other. So if we look back now with this lecture and the previous lecture, and we look at all the eukaryotic cell structures, we really can probably group them into four groups based on what they do for the cell. The first are those that are responsible for genetic control. So this is going to be your chromosomes, your nucleoli, in part the nucleus, because it's holding the chromosomes, partly the ribosomes, because they're going to be building proteins later that are coded for in the nucleus. The second group are our endomembrane system, which is involved in manufacturing, distribution, and breakdown. So this is going to be the nuclear membrane. This is going to be the rough and smooth endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, vesicles, and vacuoles. And then there's the two organelles associated with energy processing, mitochondria and chloroplasts. And then finally, those structures associated with structural movement, support, and communication, which are going to be the cytoskeleton. That's going to be the centrosomes with their centrioles. This is going to be the cell wall, the glycocalyx, the junctions in animal cells, and the plasmodesmata in plant cells. And here's a table that you can refer to that kind of gives you a small visual to remind you of an organelle or structure. It gives you an idea of what its functions are, and then it identifies whether it's found in just plant cells, just animal cells, or both cell types. So in terms of review material for the exam, these are the key terms that you should know from this part of Chapter 3. And these are the key concepts that you'll want to study when you prepare for the exam.